Hi and welcome to my OCRA A-Level Biology revision session with me, Christine. So today's lesson I want to look at B lymphocytes and specifically the antibody production. So let's take a step back and just remind ourselves about the biological membranes of cells. Now we know we've got our phospholipid bilayer. We know that we have got proteins which are embedded in that phospholipid bilayer. So it's important that we note that we actually have receptors, and receptors are very specific to the molecules that are going to bind to. So our B cells have got these unique receptors that are specific to antigens, and these receptors have got the same shape as the antibodies that we're going to talk about. So it's important that we note that the antigens are either part of the pathogen cells, or we also can call toxins that are produced by the pathogens antigens. So when we have a antigen that is specific to our receptor, what's going to happen is this antigen is going to bind. They will have a complementary shape, which means that they will form bonds and they'll be able to bind together. Now, once the antigen has bound to that receptor on the B cell is then going to process that. And what it means when it processes it is that it's going to take it in, it's going to cut it up, and then it's going to present some of those peptides on its surface. So what we have is the B lymphocyte is now becoming a antigen presenting cell, because what it has is an MHC complex. Now, because it's got an MHC complex, what's going to happen is our T helper cells are going to recognize this polypeptide on that antigen presenting cell, and that's going to result in this T helper cell releasing cytokines. Now those cytokines, those chemical signals, are going to now activate this B cell, and this B cell becoming fully activated means it's now going to go through this rapid division by mitosis. So this is where we have that clonal selection, the B lymphocyte that has the complementary receptor to that antigen being selected for, and clonal expansion, where this mitotic division, this rapid division, clonal expansion of the cells is going to occur. So we know our B lymphocytes then are going to go through mitotic division, and we know that they can differentiate into plasma cells and memory cells. What we're really interested in today's lesson is these plasma cells and the fact that they can produce antibodies. So again, I'm going to take you a step back and remind you about Module 2. So we learned in Module 2 about protein synthesis. We learned that inside the DNA we will find our genes, our short sections of DNA which code for our proteins. Our DNA will be transcribed onto our mRNA. The mRNA contains those codons which will then be translated into the polypeptide, the amino acid sequence which makes up the primary structure of our protein. Now remember that those amino acids are going to be held together by peptide bonds. And I want to note here that cysteine is the only amino acid which actually contains the sulfur atom. And that's important when we look at how the structure of the protein folds into the shape that is necessary for the antibody. So let's just remind ourselves, we have our primary structure, which is our sequence of amino acids. We then look at our secondary structure. The interactions, because of that primary structure, is going to result in weak hydrogen bonds between carboxyl and amine groups. Now that will either be alpha helix or it will give us a beta pleated sheet. That will then continue to fold into the tertiary structure, and that folding and bending, those intermolecular bonds and interactions of those R groups are going to cause this shape, which is the important thing when we look at our antibody structure. Just a reminder, there are 4 you need to know when it comes to tertiary structure: ionic bonds, hydrogen bonds, hydrophobic, hydrophilic interactions, and disulfide bonds. Now remember, I said the cysteine is the only amino acid that contains the sulfur atom. Therefore, wherever there are two cysteine amino acids, that is where you're going to end up with a di-2-sulfide bond. And then last, reminding ourselves that our quaternary structure is our interactions between two or more protein subunits. So let's have a look then at our antibody structure. So we can see quite clearly there are some key areas we need to note. We have our antigen binding site. So this is the site that is complementary to that antigen. So remember, the fact that it's complementary means that its shape needs to complement that antigen. That means that this region here within the antibody is the only variable region. There are many different antigens, therefore we have many different antigen binding sites, and in all the antibodies, this is the only section which will have a different sequence of amino acids. That is known as the variable region. The rest of the antibody structure is called the constant region. So that is exactly the same. It is always constant, and it has the same sequence of those amino acids. Now, because it's got the same sequence of those amino acids, what's going to happen is that folding, that bending structure, that tertiary structure that we have got, because this is a globular protein, immunoglobulins, these are globular proteins, they therefore have a globular shape. That therefore means that they have a tertiary structure. Now it's important that we note that we don't just have one polypeptide chain here. In our amino acid structure, we actually have 4 polypeptide chains. We have 4 polypeptide chains which are held together in a very specific shape, which have got these antigen binding sites and also receptor binding sites. And the reason it's made up of 4 polypeptide chains is because we have 2 which are called the light chains, they are much shorter, and 2 which are the heavy chains. So there are 4 polypeptides, 2 light chains, 2 heavy chains, and they are held together by these disulfide bridges. So we have a disulfide bridge between our 2 heavy chains, and we have disulfide bridges between our light chain and our heavy chain. Now we also have a hinge region, and that allows for movement, so that when one of the antigen binding sites has bound to a pathogen or to the antigen, the toxins, there is flexibility within that antibody structure, and that therefore allows for the role of the antibody to continue. So let's have a look then at how these antibodies are actually produced and secreted. So reminding ourselves, we had transcription that occurred inside the nucleus, translation that occurs at the ribosomes. Now reminding ourselves that these antibodies are going to be secreted into the bloodstream. So because they're secreted into the bloodstream, that tells us that these antibodies are produced at the RER, the ribosomes that are attached to the endoplasmic reticulum, so our rough endoplasmic reticulum. So the polypeptides that are being produced are going to be placed into RER vesicles. They are then going to be moved by the cytoskeleton to the Golgi apparatus for the further modification and packaging, and they will be packaged into secretory vesicles, which will then move to the plasma membrane and will then fuse, and exocytosis will occur, and that will expel that protein. So the B cells, the B plasma cells, which are going to produce and secrete these antibodies, need to go through that process of transcription, translation, and then secretion. So make sure you're always putting those links together. Okay, so we have our antibody structure then, and what's important is that we understand that there are different types of antibodies that have different roles to play. So we have the ones which are antitoxins. They will bind to the toxins to neutralize them. Remember that antigen binding site that's specific. If it binds to the antigen, it stops it from able to cause any harm. We have agglutination. We have the fact that the antibodies can actually stick to the pathogens. Now, remember I said about the hinge regions, that allows for multiple pathogens to be bound to the same antibody and therefore clumps them together. And the other thing that they can act as is opsonins, and if they're opsonins, their opsonization is going to occur. What we're saying is that receptor binding site is going to make these pathogens more recognizable to those phagocytes. So it's important that we know the three roles in which the antibody plays. But let's just take a step back and remind ourselves that those B lymphocytes are going to differentiate into not only those plasma cells, which are going to produce and secrete those large amounts of antibodies into the bloodstream, but also to produce memory cells. And when they differentiate into the memory cells, these are the ones that are going to remain in the body for longer periods of time. And what that means is when you have a secondary infection by the same pathogen, what you're going to get is a quicker response. And that's because we have already had the clonal selection and clonal expansion occurred. So if we look then at our antibody concentration in the blood, and they love to give you a graph that looks like this, the key thing to note is that our first exposure is where we get that clonal selection occurring, and then we have clonal expansion. That clonal expansion will result in the mitosis and differentiation of cells to produce lots of antibodies. Therefore, our antibody concentration is going to increase in the blood. Now remember, if those antibodies are now sticking to the toxins, or they're clumping the pathogens together, or they are making it more recognizable, what's going to happen is the phagocytes are going to come along and they're going to engulf and digest the pathogens along with the antibodies, and therefore your antibody concentration in your blood is going to decrease. So the first time you encounter the pathogen and you have that response of your immune system, that is known as your primary immune response. The second time you have the exposure, because you have those memory cells, they're going to allow for a faster response. So clonal expansion is going to occur again, and then the production and secretion of these antibodies. And because we have more memory cells, it means that that expansion is going to happen a lot faster. Therefore, more antibodies can be produced, and that allows for that big increase in the antibody concentration in the blood in a much shorter amount of time. So that is what's known as your secondary immune response. But it's really important to note that, like any other cell, your cells will not last forever in your lifetime, and therefore your memory cell numbers are going to decrease over time. That is a natural part of aging. So you will see that your antibody concentrations will decrease because your phagocytes are going to engulf and digest, but you're also going to have a reduction in your memory cells as time goes on. And so therefore you will need to give your body time to build up that immunity again. So I hope you've liked this video. If you have done, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, please do check out my revision platform, www.aiqchat.com.